Let's start with something we often take for granted: our medicines. Millions of people rely on routine medications every single day. But when we hear about drug recalls due to impurities like nitrosamines, it raises an important question. How much of these substances is actually dangerous, and how much is it still considered safe? In this presentation, we'll explore acceptable intake limits for common nitrosamines and how science helps us balance medicine safety with unavoidable trace contaminants. Hi, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories Canada. Welcome to Resolve Mass Laboratories. We bring deep scientific expertise in advanced analytical characterization and complex molecule development to accelerate your IND and ANDA programs. Now, what are nitrosamines? So first, what exactly the nitrosamines are, what these impurities is. So nitrosamines are chemical compounds that form when amine react with nitrites. These happen during manufacturing, storage, and even in everyday processes like cooking food and many more. They are not something new or unexpected. We actually encounter them in small amounts in food, water, and even air. However, some nitrosamines are classified as probable human carcinogens. But here is the key idea. Risk doesn't come from just their presence, it depends on the dose. So the big question here is, how much is the safe? Because this brings us to the most important questions of all these nitrosamines subjects, and that is, how much is actually a safe limit for these impurities? In an ideal world, we would eliminate these compounds completely, zero. But in reality, trace levels can sometimes be unavoidable. We cannot really avoid them. So instead of asking, is it present? Scientists ask, what level can a person safely consume every day over a lifetime? And that's where the concept of acceptable intake or AI comes in. What is acceptable intake? Acceptable intake is defined as the amount of a substance that can be consumed daily over a lifetime without significant health risk. It is measured in nanograms per day or else ng per day. Now to put that into perspective, 1 nanogram is 1 billionth of a gram. So we are dealing with extremely small, tiny quantities. These limits are intentionally set very low to ensure long-term safety even with continuous exposure. Now, you might wonder, how do scientists actually calculate these limits? How regulatory authorities have defined these acceptable intake limits for each nitrosamine impurities published for now? So it's a multi-step process. First, animal studies are used to determine something called TD50, the dose that causes tumors in 50% of test animals. And then mathematical models are used to extrapolate this data to predict human risk at much lower doses. Next, large safety factors, often around 100-fold, are applied to account for differences between species and individuals. Finally, the limits are set to ensure that the lifetime cancer risk does not exceed about 1 in 100,000 people. So these numbers are not arbitrary, they are extremely conservative and science-based. So, common nitrosamine limits. Let's look at some real examples. For example, NDMA, one of the most commonly discussed nitrosamines, has an acceptable intake limit of 96 nanograms per day. It has been detected in drugs like ranitidine and valsartan. On the other hand, NDEA has a much lower limit, 26.5 nanograms per day. Why the difference between these two? Because NDEA is more potent as a carcinogen. So even though both are nitrosamines, their acceptable limits differ based on their toxicity. Now, let's further talk about some common nitrosamines limits. And another example apart from NDMA and DEA is NMBA. This is also a common nitrosamines present in many drug components and other materials. And NMBA, which has a limit similar to NDMA, 96 nanograms per day because of comparable potency. This highlights an important principle. Stronger carcinogens are assigned lower intake limits to maintain the same acceptable risk level. Also, acceptable intake is not determined by the chemical family but by the individual compound's toxicological profile. So each nitrosamine is evaluated independently. Now let's talk about how nitrosamines actually end up in pharmaceuticals. How they form in pharmaceuticals. So they can form during manufacturing due to impurities in raw materials or reactions during the synthesis. They may also come from packaging, for example, interactions with adhesives or container materials, and sometimes even during storage. Factors like heat, humidity, or time can lead to degradation and formation of these kind of nitrosamine impurities. So it's not always a single cause, it's often the combination of many factors. Now, what regulators are doing, what regulatory authorities are monitoring in the area of nitrosamines? Regulatory agencies like FDA and EMA have taken several actions. They have established strict acceptable intake limits for various nitrosamines. They require pharmaceutical companies to test their products and monitor risk. And importantly, they try to balance safety with availability, because removing essential medicines from the market can sometimes create bigger health risk. So it's a careful balance between minimizing risk and ensuring access to treatment. This brings us to the end of the session, and let's conclude practical takeaway from this session. So what should we really take away from all of this discussion? First, zero risk is not always realistic, but controlled and well managed risk is achievable. Second, acceptable intake limits are designed with large safety margins to protect public health. And finally, strong quality control, deep process understanding, and advanced analytical methods are critical in preventing contamination. At the end of the day, ensuring medicine safety is about understanding the science and applying it responsibly. If you are navigating complex drug development challenges from IND enabling studies to ANDA submissions, connect with Resolve Mass Laboratories to accelerate your program with confidence. Thank you so much for watching this session. See you in the next one. Bye for now.